Okay, so I'm going to take you through a few different stories today. First, starting off telling you what we're doing in Singapore. And one of the reasons I've stayed in the academic side of the world is that we really want to try to target aging and longevity directly. And so I get the fact that if you're developing drugs, you need disease indications and all of these issues. But on an academic side, we can ignore a little bit of that and try to get data to see if we're really influencing the aging process or not. So we have a couple programs looking at healthy longevity at NUS, and it's really a full pipeline now. There are about 35 faculty working in these programs, everything from preclinical research to translational, and really I think an advantage we have is we can focus on clinical science. So I'll tell you a little bit about how we're doing those kinds of interventions to target longevity. And we're also trying to promote longevity science both in Singapore and globally. I'll tell you a little bit about that. A lot of what we're doing academically, I don't think anyone is completely unbiased, but we're trying to be as agnostic and collaborative as possible. So if you have interventions, either academic or private sector side, that you want to test in various animal models or humans to see if it targets biomarkers of aging, we're happy to work with you on that. And we have a few company partnerships already. And so on the preclinical side, I won't spend much time on it, but we have pretty much the full suite of animal models, except for maybe primates. We're doing non-vertebrates, really interested in killifish studies lately. We have 2 faculty members working on that, and some positive hits for some of your favorite longevity molecules. Stem cell models, organoids, things in that area. A lot of mice, I'll tell you a little bit about that, and we have a lot of AI people now working on bioinformatics, which I won't spend too much time on today. I think one of the things we're doing on the human side, besides looking at interventions, is to look at biomarkers of different types. And so looking at systemic biomarkers of aging, and you've heard about things like epigenetic clock and others. We've been working on a lipid clock lately. But also markers of each of the hallmarks of aging, because one of the challenges, we don't know how markers of hallmarks of aging line up with systemic markers of aging and how they line up with organ and tissue-specific markers of aging. So if we can look at all of these in the same samples, we can really begin to sort of connect the dots here, hopefully, to tell us a little bit more about what these systemic biomarkers are teaching us. And we've altered our mouse approach pretty dramatically now, where I'm going to show you one little mouse thing today, but mostly what we're doing is looking at 6 to 9 months interventions in mice starting around 18 months of age, and using frailty and biologic aging biomarkers as outcomes in the mice rather than survival. It's not that I don't think survival is the best assay, it probably still is, but it takes a long time and it doesn't align with the human studies. And so what we're interested in is how do we make the mice aligned to humans and predict the outcomes in humans as much as possible. And so we've altered how we do mouse aging experiments. Happy to talk about that. On the human side, we're really interested in validation. How do we take the huge number of interventions that are likely to affect aging, or at least they do in animal models, and see which ones work in humans? Everyone has their favorite argument for why their intervention is going to work in a human. I can give you mine too. But we really don't know, I think, which ones are going to have the biggest effect. So it's really by comparing and contrasting them in smaller studies that we can begin to tease out which ones are more likely to work. I think ultimately we need to personalize that too. We need to understand which interventions are going to work in which people at which time. It's very early days in that process, and we're doing studies like that both in humans and mice now. We have a mouse longevity spa, if you know any mice that want to come, where we're trying to personalize interventions to slow aging. And then we need to, and at least on the academic side, we need to be able to scale. So Singapore is giving us a lot of money to look at healthy longevity. We need to be able to measure aging in the population for a reasonable price, and then develop interventions at a reasonable price that we can scale to an island. And so we're very mindful of how we do that as we go forward. I should point out that Andrea Meyer in the picture has joined the team at NUS, and she's really my co-partner in crime now, and has an extensive amount of clinical experience, so it complements my preclinical experience, so hopefully that'll get us moving much faster. On the human side, we're really using biomarkers as endpoints, not Not that we think the FDA is going to buy into that immediately. We don't really care. The point is to get validation that things are working. And so we're doing 6 to 9 month studies in relatively healthy people. I'll tell you a little bit more about that, using a wide array of biomarkers, and not just the AI-driven ones, but physiologic parameters, things like pulse wave velocity, grip strength, VO2 max, and a number of the more classical biomarkers of aging. 45 to 65-year-olds, and we're comparing and contrasting a wide range of different kinds of interventions. Already we have a cross-sectional study nearly completed on 450 Singaporeans, deeply phenotyped and across ethnicity in Singapore, and we've collaborated with the National Precision Medicine team in Singapore to look at 10,000 genomes, both GWAS and DNA methylation, as well as telomeres. And they're interesting. I can't talk about that data, it's a big consortium, but there are interesting things that are going to come out of that with regard to rates of aging in different ethnicities in Singapore. Like I said, the human intervention studies are going, and we have a wide range of collaborations. So if I want to take a step back, I've talked a lot about how can we target aging directly, that only matters if it's feasible to do that. I think this audience is probably convinced it is feasible, or you wouldn't be here. But it's worth pointing out, and this is something Eric mentioned yesterday as well, that there are a wide range of interventions that have been proposed. If you look at the drug age database, there are over 1,000 distinct drugs that have some impact on longevity in some animal model, over 100 in mice. And I think that's even more of a call for us to begin to test these things more in humans, because we have no shortage of interventions in some respects, although in another respect, many of these interventions are targeted to the same pathways. And so I want to come back at the end of the talk and say that we do need to increase the universe of longevity interventions, but in a new way. In any case, we work on all of these things on the left, and I'll tell you about alpha-ketoglutarate and Gemfibrozil a little bit. But the other ones in green, we have at least interesting data in mice and/or killifish that they affect aging. So one of the good things about this is that most of these drugs we didn't begin studying. They've been studied in mice by other people, and we see a pretty good rate of retesting. So that's, I think, giving us confidence that some of these things are at least likely to work in mice and may work in humans. So this is, I'm going to just give you some human updates on the studies sponsored by Ponce de Leon Health, which I'm involved with, full disclosure. So this started with a collaboration with Gordon Lithgow at the Buck Institute to look for natural products that impact worm aging, and specifically combinations of natural products, because for a company based on supplements, it was possible to get IP around combinations. So I should tell you, I'm going to tell you 3 quick stories. One is supplement, one is a drug, and one is a screening platform. And I'll try to go fast through them. This product is now on the market. There's a product called Rejuvenate, which has alpha-ketoglutarate, time-release format, plus low-dose vitamin A if you're a man, or low-dose vitamin D if you're a woman. And it came out of the studies originally in worms and some of the mouse studies that Gordon and I did, starting at 18 months of age, doing interventions. In that case, we were looking at survival of the mice, but also frailty and a range of other parameters. This is published a while ago, so I'm not going to talk about it, except to show one slide, which is the frailty slide. So this is Susan Howlett's frailty matrix index. Sorry, I'll get the word right in a minute. And you can see that the control animals in blue become more frail. That's the higher you get on the frailty index than the alpha-ketoglutarate treated animals. This is just AKG in mice. And if we normalize that, we can see about a 50% reduction in frailty in both females and males. Now, the effect on lifespan is much smaller than that, maybe 5 to 10%. So really what we argued from this paper is we were compressing morbidity in mice. But again, this is mice, and we wanted to go to humans. So we published one study last year, and I just want to give you all the qualifications on this one. It's not placebo-controlled. This was users of the Rejuvenate product that were willing to take methylation tests. And so it has all the problems you get from that kind of a study. And in fact, I'll just say right now that I think if you're paying $100 for a product to be younger, you're probably younger. I think that there's probably a small placebo effect. We've seen that in other contexts, and if you believe these biomarkers as indicators of age, there is a small effect. So when you're looking at a study like this, you're probably combining placebo plus hopefully the interventions effect. These are people at a late middle age range, 14 females, 28 males, an average of 7 months of taking the Rejuvenate product. And what you can see is we saw a big effect on biologic age using this TrueMe methylation test. This is a simpler methylation test. It's a saliva-based one. At the time, that was the only saliva-based one available. And so just to let you know, basically what you see here is on the left, if the arrows are going up or the direction is going up, that means people are getting biologically younger by this test. It was about a 7-year effect in a 7-month taking the product, which is a big effect. What made me tend to believe this is we saw 2 strong correlations. And this is one point I want to make, and I think it may be true of other longevity interventions as well, though we don't know for sure yet. You might have expected that people taking AKG, the younger people that were actively exercising, would get the biggest benefit from it because it is used as an exercise mimetic. What we saw, however, was that people that were chronologically older got the biggest benefit, and also people that were biologically older than their chronologic age got the biggest benefit. So it was the people that were not biologically young, the couch potatoes, if you will, that were getting the biggest effect. So that made me think there might be something to this study, even though it wasn't particularly controlled. And that led the company to go back at Indiana University and do another study that was placebo-controlled. And this is one of the reasons I'm telling you this, is we learned how not to do studies. So we decided to this was before we had the data from the previous study, we decided to take 45 to 65-year-old people without disease, when it happened to be during a pandemic. And when you look at their biologic age, and this time by the Levine clock, the blood phenoage clock, so we use multiple clocks in this study, those people were about 6 years younger than their chronologic age. So if you take people in Indiana that don't have disease in that age range, they're by definition biologically younger. And that may be augmented by the fact that they're willing to come into a hospital for a participant-based trial in the middle of a pandemic. But in any case, we selected about the youngest crowd we could find, and I just showed you on the last slide, those are the people that don't respond very well. So if you compare to placebo, we have a trend, but not significance. But if you look at this Levine phenoage clock, there's an interesting result that comes out of it. So this is the blood markers that are used for the clock. Mostly things you get in a complete blood count, and nothing particularly unique here. And you can weight them differentially and get a phenotypic age. And again, what we see is the response is by people that are biologically older. And so what we're looking at here is that on the bottom axis, I don't know if you can see the arrow, good. I can even make a laser pointer and get technological here. On this axis, you're looking at their biologic age minus their chronologic age. So if that's negative, that means they're biologically younger. So you can see that most of the people that came into this study were about 6 years on average biologically younger than their chronologic age. This is the change in Levine clock age by visit. So this is 6 months. So every 3 months, they get this much different in the phenoage clock. And again, if you look at people that are within 5 years of their chronologic age or older, they all respond. And so I think what we're seeing is something similar to the last study. And so what we're doing now in Singapore is doing just time-release AKG because we want to simplify mechanistically what we're studying. And as inclusion criteria, we're going to take biologically older than your chronologic age to get into the study. And so I think the point I want to make is that we still think this is likely to work in people that are of the normal age range or biologically older. But my main point is we still need to think about how to do these studies. And how do we set inclusion criteria? How do we study longevity and aging and not diseases of aging in a way that makes sense? And so we're learning as we go. I want to talk briefly about Gemfibrozil. This is a drug I also started working on at the Buck Institute, but we've expanded it a lot now. And what we found is that through some weird correlations with yeast, we found that it extended lifespan in yeast. It also extends lifespan in worms, and it extends health span in parameters in worms as well. We never published that, but we got interested in the drug. And so what is Gemfibrozil? This is a member of the fibrate class, widely used. It was introduced by Park Davis in 1976. And it's really to reduce triglyceride levels. So this was one of the initial studies, and people taking Gemfibrozil had a reduction in triglycerides and an increase in HDL cholesterol. It's a relatively simple molecule. And so we're wondering why it was affecting aging. And so the mechanism that it's supposed to act through as a fibrate is activation of PPAR alpha. And so it gets into the cell and it disrupts a complex that contains repressors and the transcription factor PPAR alpha, frees the transcription factor to go to the nucleus and regulate a number of genes involved in lipid metabolism. It all sounds great, except we don't believe this is the mechanism for Gemfibrozil for a couple reasons. One is that we don't see activation of PPAR alpha in our hands. Another is that if you knock out the PPAR alpha ortholog in yeast and worms, that Gemfibrozil still extends lifespan, there's no effect. And the third, and perhaps most telling, is other fibrates, which have much stronger effects on PPAR alpha, don't affect lifespan in our hands. So we're trying to figure out what this drug really does. One of the clues came from going back to yeast. So I'll show one slide in yeast, and you can do cool experiments in yeast. One of which you can do is ask yeast to grow on 100 different carbon sources, and then ask where the drug sensitizes the growth. And what we found is that Gemfibrozil had no effect unless you tried to grow yeast on dipeptides and tripeptides as carbon sources. And so that suggested that there was something going wrong with peptide metabolism. And in fact, if we add the drug to the yeast cells, we could find a reduction of about 30 to 40% in levels of almost all the amino acids in the cell. And you can also do elegant experiments like this one where you add a toxic dipeptide that when it gets into yeast, so it diffuses off this paper, it kills the yeast. But when you add Gemfibrozil, you can block that, meaning that this toxic dipeptide is not getting into the yeast cells in the presence of the drug. So we could go back in worms and show that we could block fluorescent dipeptides from getting into cells through the intestine with Gemfibrozil. We can look in human cells and show that we can reduce amino acid levels with Gemfibrozil. And maybe this experiment is one of the most telling. We can actually take the intestine of mice ex vivo, put fluorescent peptides in it, and then add drugs and ask what happens. So this is vehicle, you get absorption of the fluorescent dipeptide. If a positive control is losartan, which is another interesting molecule that we might think of for aging, and then Gemfibrozil, they both block amino acid import. So we think we have the target for this, and we're actively studying that. And if you block amino acid import into cells, the downstream effect of that is going to be inhibiting the mTOR pathway. So one of the downstream benefits of Gemfibrozil is through reduction of mTOR, although we don't think that's the only thing. And this is an elegant way of showing that. You take cells in culture, look at phosphorylation of a target of mTOR, S6 kinase. If these cells are starved, they have very low phosphorylation. Gemfibrozil does nothing. If you add single amino acids, you can activate TOR, and Gemfibrozil does nothing. If you add leucine, you can activate TOR, and Gemfibrozil does nothing. And if you add a dipeptide mix, you activate TOR, and it's blocked by Gemfibrozil. So I think this is a pretty clean result. And what it's telling us is that we can modulate amino acid uptake with this drug, which is a relatively safe drug, especially as fibrates go. And so we're really looking at expanding that. We have three targets now for Gemfibrozil, one of which is involved in amino acid metabolism, and the other two are involved in lipid metabolism, and we think those are the real targets for the hypertriglyceridemia. So what we're looking to do now is to spin out a company. We're already looking at derivatives of Gemfibrozil so we can optimize relative targeting of these three different targets for either aging or different disease indications. So that's something I'm happy to talk about if people are interested. And I want to go on to one other small company we just started, and this is with Matt Kaberlein, who's one of the leading worm biologists and well-known in the aging field and a long-term collaborator of mine, and Jan Gruber, who's looked at many different combinations of drug targets in worms in Singapore. So Those are the two faculty founders. I'm also a founder. I guess I'm just the sommelier for this company or something. But I'll tell you a little bit about it. Two of Matt's postdocs are really running the company. One is Mitchell Lee, and the other is Ben Blue. Ben is an AI expert too, and so there's a big AI component to this. And so it's taking a step back, and it's asking the question of the landscape of potential interventions that we've screened that might affect aging. How much of that have we actually looked at? And if you look at those 106 mouse longevity drugs, a lot of them target the same pathways. And our vision is that by using sort of human intelligent design, we've picked a lot of molecules that may affect aging, but we've also biased our selection. If we go back and take a completely unbiased approach, what can we find? And how many different pathways are out there we don't know about? And we wanted to do this in organisms, not in cells and culture, because cells and culture, you have to guess at which aging phenotypes you're looking at. And so we chose to do worms. And so what Matt's done, and people at the Buck and other institutes have developed different kinds of these things. Matt's developed what he calls a worm bot, which gives us access to really high throughput screening in worms for longevity or any phenotype really we can look at. And so one worm bot can do 2,500 experiments a year. It's based on serial photography of the worms, so you can monitor the activity or movement or fluorescence of the worms if you like. And so what we've done is started a company to do really high throughput screening. The idea is to have 40 of these worm bots. We're already in the process of generating this in Seattle. And so we can screen a lot of different phenotypes. And so I just want to give you one example of one of the things we found from this. So we took the FDA-approved drug library, just as proof in principle, with just one of these worm bots. And we used an amyloid beta model for aggregation related to Alzheimer's. This model causes rapid paralysis, and so we could take different conditions. So instead of just screening the FDA library to find single hits, we were really interested in combinations. And so I want to talk a bit about combinations, especially with regard to, let me take a step back and talk about humans for a second. Many people out there are now taking 10 or 15 different kinds of supplements to enhance their longevity. I have no idea what you're doing if you're doing that. If I study this in mice, even if we start combining two things together, and we've done a lot of this now, I can't predict the outcome. So I don't want to discourage people from out there looking at supplements to affect aging. I'm just saying that once you go beyond one thing, you've gone from being on the edge to off the edge. I have no idea. There's interactions between these supplements. And actually, if we combine them together in mice, it's more likely things will cancel each other out than it is that they'll be additive. So just keep that in mind. But we think that's even more need so that we can look at multiple combinations of things that might affect longevity. And we can do this rapidly going back to a simple system like worms. So here we took control metformin, which has a tiny effect in this paralysis assay. Drugs alone are combined with metformin. And this gives you an idea of the odd effects you get with combinations. So red is the metformin alone with different combinations. Blue is the FDA drug alone, and some of them enhance paralysis and some of them reduce paralysis. And then the combinations in green. And so you can see almost anything you want out of this. So you can find things where the effect of the compound alone is almost zero, but when you combine it with metformin, here, that's a good example, you get a huge enhancement of the effect. And so this combinatorial space has really not been screened at all, and that's one of the things we really want to do. This just gives you an idea of what's going on. So we're looking at these worms moving around. Here's control, the drug alone, metformin, and the combination, and this is the relative survival curve you get. When these worms get paralyzed, we're circling them in red. And I think you can see that while there might be small effects of the drug or metformin alone, these guys that have the combination are still very active. So this is just one example of a combination that has a synergistic effect on this assay. So we have about 10 or 15 disease assays as well as looking at longevity alone. And we're just going to scan the universe as rapidly as we can, as many different libraries as we can, and try to find new compounds that have effects in aging and also disease, but also combinations thereof, like this one. So I can't tell you what the drug is yet, but there's a lot. This is really just an example. We've already have lots of hits like this, and we haven't even really gotten started yet. So that's taking a step back and doing the screening to find new things. So if anybody's interested in that, let me know. This is just looking at the aggregation and quantifying it to show the combined effect of 3 different hits compared to metformin alone. And so there's multiple drugs that combine with metformin, but that's just, like I said, one combination. I just want to close by saying that we have also, in addition to a longevity program in US, we have an Asian Center for Reproductive Longevity and Equality. This is funded by the BioEcho Foundation. They also set one up at the Buck Institute. And we're very excited to move forward this. We're looking at ovarian aging, everything from a stem cell-based approach to animals. And we have an IVF facility that's very active at NUS. And so we're beginning to look at human studies to try to preserve fertility and also better understand the biologic changes that happen after menopause that affect female aging. And so if anybody's interested in that, I can talk about it as well. So with that, one more thing, sorry. We have a healthy longevity webinar that many of you know about already. You've already told me about it and that you are watching it, but if you don't, you can go to this website. It's free. It's Thursdays at 7 in Singapore, and we have speakers from all over the world. Every one of them from the United States complains about having to do the show live at 4 in the morning, but some of them still do it. We have about 500 viewers live. It's really designed to combine scientists, clinicians, investors, interested public to educate everybody about longevity. These shows are also all available on YouTube for free, and there's 65 of them now. So you can go and find your favorite speaker and listen to them tell you what they think about healthy longevity. If you have trouble finding this, let me know. We'll make sure you get access to it. So with that, I have a lot of collaborators that are helping out. And thanks a lot. I'm happy to take questions.
Thank you. So one might